KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.com.
Students, now in this video we are discussing the nature of a bonding linking monomers in polymer. So already in previous video continuation information only we are discussing. So in previous video somewhat information we have discussed related to the nucleic acids. So in this video continuation only, nucleic acids information only, continuation information we are discussing. Why? Because in previous video we could not discuss the complete information of nucleic acids, that's why continuation. Now nucleic acids exhibit a wide variety of secondary structures. So secondary structures, there are different types of secondary structures, wide variety of secondary structures we can observe in nucleic acids. Okay, so for example, one of the secondary structures so exhibited by the DNA is the famous Watson and Crick's model. So Watson and Crick's, they have been proposed. So double helical structure of DNA, so double helical structure of DNA model they have been proposed. So this one is very famous one. I think so we have studied this one even in 9th standard and 10th standard, so that particular structure only. So here one structure also we have drawn now, so that particular structure you can see. Okay, so that is one of the secondary structures of DNA. So nucleic acids we took, our knowledge point of view, nucleic acids two types are there. One is DNA and one more is RNA. So one of the DNA structures, a very famous one, is nothing but double helical structure of DNA proposed by the Watson and Crick's model. So this model that says that DNA exists as double helix. So DNA, according to this particular model, DNA exhibits or exists as double helix. Double helical structure already we seen this one. Next one, two strands of polynucleotides or run anti-parallel to each other. This is important point. Now here two strands are there. So these two strands of DNA runs anti-parallel to each other. Anti-parallel means, once you see the diagram, so one strand is running from the 5' to 3' direction. So again another strand it is moving from the 5' to 3' direction in opposite way. You see, one strand mele to keela vandachu and one more down to up. So thereby here we have to see a 5' to 3' direction only. Okay, so 5' to 3' direction and here 5' to 3' direction. So thereby, so what are the strands are there in DNA? So these two strands runs anti-parallel to each other. That's why here we have seen that is runs in opposite directions. Run opposite direction. Okay, next one, the backbone. The backbone is formed by the sugar and phosphate and sugar chain. So means here we I told you, so backbone is definitely formed between the phosphate and sugar, isn't it? So thereby here what is the main backbone? Here we are calling by one more name, phosphodiester bond. Phosphodiester bond is formed between the sugar or simply pentose sugar and phosphate. Don't forget. So thereby here, up to here, what are the points we seen? So in nucleic acids, some kind of secondary structures or wide variety of secondary structures are there. Best example is Watson and Crick's model, if you observe now there, so DNA showing that double helical in nature. So there two strands of polynucleotides runs anti-parallel to the each other. So anti-parallel to the each other means here the strands which runs opposite direction, which run in opposite directions. And here if strands, suppose here these are all running anti-parallel to each other, no, so there suppose if you take each strand, it is made up of three different types of components. So in previous video I think so diagrammatical explanation I have given, no, so there nucleotide and nucleoside information we learn, no. So one nucleotide is nothing but, or one nucleotide is formed by the three components. So there phosphate, pentose sugar and nitrogen bases, isn't it? Ipo phosphate and pentose sugar and nitrogen bases, these three components comes together and involves in the formation of nucleotide. Emma ipo backbone bond means here the phosphodiester bond formation taking place between the phosphate and pentose sugar. Okay, so now here nitrogen bases. So our knowledge point of view, nitrogen bases, what are the things are there? Now nitrogen bases, in previous video we have learned now two types of nitrogen bases, purines and pyrimidines. Purines are adenine and guanine we took, and pyrimidines, so remaining three means cytosine, thymine and uracil. So thymine is present in DNA and uracil is present in RNA. Emma ipo nitrogen bases projected more or less perpendicular to the backbone but face inside. Emma, so here diagrammatically diagram also is there, I will show you now. So but information ipo save பண்ணிக்கோங்க. அது என்னன்னா nitrogen bases are projected more or less perpendicular to the backbone. So backbone కి nitrogen bases arrangement how it will be like a perpendicular more or less means 90 degrees angle madri will be there and the face will be inside. So the structure point of view that will be inside. Okay, diagrammatically I will show you just 5 to 10 minutes of time. Now here what is the information? Now nitrogen bases, if you see adenine and guanine of the one strand compulsorily base pairs or base pairs with the thymine and cytosine respectively. So means once you see adenine always binds with thymine and guanine always binds with the cytosine. Once you read this statement, adenine, A indicates adenine and G indicates guanine. So of one strand definitely or compulsorily base pairs with the thymine and cytosine. Appadena adenine with thymine and guanine with cytosine. That's why here we have written respectively. Okay, so now without to avoid the confusion, here we have written like this: adenine binds with thymine and guanine binds with cytosine. So here of the another strand means here suppose in one strand adenine and guanine or adenine is there and opposite strand nalli yaar irukku thymine and next strand nalli G is there means and opposite strand nalli C will be there. So that is also I will show you in the diagram. Next one, there are two hydrogen bonds between the adenine and thymine. So there are two hydrogen bonds we can see between the adenine and thymine, and three hydrogen bonds are there between the guanine and cytosine. Even this point also is important. That's why here we mentioned once you see here between the adenine and thymine we like is equal to is madri is there, no, adi two hydrogen bonds, and between the guanine and cytosine no, three lines are there, no, adi indicates the three hydrogen bonds or the presence of three hydrogen bonds. Amma puriyudha aprom each strand appears like a helical staircase. So one by one you see there. Suppose if you take each strand, no, whether here beside to this diagram, no, one more diagram is there. Whether that is visible or not, I don't know, but I'll give you that particular diagrammatical explanation. So here each and every structure or each strand appears like a helical staircase. Each step of ascent is represented by a pair of bases. So here definitely suppose each and every step if you see, so here it is seems to be like a base pairs. So at each step of ascent the strand turns 36 degrees angle. సో యాంగిల్కి మేలే 36 మేలే చిన్న డాట్ లైక్ మొట్టమొదటి రీజన్ చిన్న జీరో అదిదా సో 36 degrees. Okay, so at each step of a ascent the strand turns 36 degrees. So one full turn of a helical strand would involve 10 steps or 10 base pairs. So means, so suppose if you take a full turn means how many degrees we can take? 36 degrees, 360 degree. Why? Because here 10 steps are there, no? Emma, 10 in 36 into 10, so 360 degrees, isn't it? So which consists of 10 base pairs. So that's why here one turn if you take, Emma, each turn or each step if you take, there no 36 degrees means 10 steps over there means 36 into 10 dana. So 360 degrees angle. So next one, the pitch would be 34 angstroms. Pitch, so pitch means here the diagrammatically I will show you, the pitch would be 34 angstroms. Emma, pitch would be 34 angstroms. So one pitch or one length, and here you see, so here I have given information pitch or one helix or one turn. So now here one pitch would be 34 angstroms and the rise per base pairs would be 3.4 angstroms. So here each base pair, idi, so one pitch nalli, so 34 angstroms distance is there means, so here each base pair so would be the distance between the each successive nitrogen base distance is 3.4 angstrom. So means here once you see the diagram, so this one I'll give you based on the diagram here. So from here to here one line is there, no? From here to here we have written this one as one pitch or once you see here one pitch or one helix or one turn, here we took from here to here, isn't it? So there that particular name is pitch or helix or turn. What is the length we took? 34 angstroms or 3.4 nanometers. Okay, and here count it, here how many number of nitrogen base pairs are there? Here we learn how many must be there? 10 base pairs must be there. So thereby here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so from here to here 10 base pairs are there. Ipo base pairs, once you listen about that one, pairs, base pairs, appadena 20 nitrogen bases or 10 nitrogen base pairs. புரியுதா? 10 if you are writing 10 no base pairs write பண்ணீங்க. If you are writing with 20 no nitrogen bases write பண்ணீங்க. pairs write பண்ணக்கூடாது 20 write பண்ணும்போது. சரியா? அது. அப்புறம் இப்போ பாருங்க இங்கே from here to here 10 nitrogen base pairs are there, no? So between the each nitrogen base pairs, now here you see between here இங்கே சின்ன arrow mark is there, no? So so from here to the here there what is the length is we are finding 3.4 angstroms or 0.34 nanometers. So that value how we have obtained? So once you see 34 by 10 poduvana 3.4 angstroms varum, or so this one we can convert it into nanometers, that is 0.34 nanometers. So actually here if you take one helix, or one pitch, no, there, so two grooves must be there. One is a minor groove and one more is a major groove must be there. So likewise only we have to draw one helix. And here one strand or opposite strands if you take there, no, both sides you should not take a 5' and a 5'. Okay, one strand if you are starting with a 5', no, opposite strand must be 3'. Why? Because we don't know, so DNA definitely both strands of DNA runs anti-parallel to the each other. Point purida. Next one, ipo again come to the information. So this forms the DNA with the above mentioned salient features known as B DNA or B type DNA. So means here we don't know. Suppose if you take a total pitch turn 360 degrees angle and 34 angstroms one pitch length, and next one adenine always binds with thymine with 2 hydrogen bonds, guanine always binds with cytosine with 3 hydrogen bonds. So all these points and the DNA DNA runs anti-parallel to each other, and 10 nitrogen base pairs are there, or 20 nitrogen bases are there. Yeah, LMA, all these points, definitely we will study under B type DNA. So by seeing all these salient features of DNA, we can recognize that particular DNA comes under B type DNA. Even our studies point of view, what are the salient features we will study? All salient features comes under B type DNA. So B type DNA only generally we will study in our classes. That is a point now I am going to give you. Next one, now you see, now diagrammatical explanation now I will give you. So now you see, so there small rough diagram we have drawn, first diagram. This one we have explained, now pitch, helix, turn, 34 angstroms or 3.4 nanometers, and the distance between two nucleotides or two successive nucleotides, 3.4 angstroms or 0.34 nanometers we seen. அப்புறம் பக்கத்தில் இதே சின்ன ஃப்ராக்மெண்ட்டை எடுத்துக்கோ, இங்கே இது கட் பண்ணி இங்கே போடுவோன்னா உள்ளே போகும். So structure there, actually this particular structure I should not give you now only, but for better understanding purpose point of view now I am going to give you. Again, don't say sir this diagram not there in a textbook, why you gave like that, you don't ask me. Right. So for just for better understanding purpose only I have drawn this diagram, so don't misunderstand. Right. Now here pentose sugar. So you see there, this is S pentose sugar, this is P phosphate, and here middle is adenine and this thymine. So with two hydrogen bonds, adenine always binds with thymine, and thymine also simultaneously always binds with adenine. Guanine with cytosine and cytosine with guanine. So for easy to understand, I have written like this. Next one, so from here to here, once you see, so here hydrogen bonds are there, two hydrogen bonds, three hydrogen bonds. This is one strand, and this one is another strand. That is on the side. Two strands, DNA double helical structure, this is one strand, this is another strand, pakatali strand. That's why here even here we have written either 5' kile 3' we wrote, and kile 5' and mele 3' we wrote in another strand. Ipo paringa diagrammatically. Ipo here pentose sugar is there, here the phosphate is there. Phosphate, here this particular phosphate you are observing, it is attached to the pentose sugar with a 5th carbon. So that's why here it is last one. So in this strand, that's why this one we will be calling as a 5'. Actually this point is there in a second year in +2. So once you listen here, suppose if you are unable to understand, even in +2 also, I will explain that particular point. Yeah, what is the point now? Ipo pentose sugar ki 5 carbons are there. 5th carbon ali CH2OH, even in previous video diagram also we see CH2OH ange, that one making bond or linked with the phosphate. Suppose this phosphate is the last one in that particular strand, that particular strand we will be taking as a 5'. And inge phosphate is pentose sugar is there. A pentose sugar's 3' OH group is there, that one is a free. Actually this OH will make bond with the phosphate also, but here free OH is there, free hydroxyl group is there, that one. So when we are observing OH group, that particular side we will be considering as 3'. And middle one, if you see there, here the pentose sugar is this one with third carbon, and this one with 5th carbon. Suppose if you take one pentose sugar, 5th carbon and 3rd carbon only linking with phosphate. That's why here we see this line this one and this one together we will be considering as a phosphodiester bond. One link if you are taking phosphoester bond we have to take, or simply ester bond we can take. Rendu together phosphodiester bond we can take. Means, listen carefully by one more time. If you take one pentose sugar, that pentose sugar consists of 5 carbons. One carbon, 5th carbon making bond with one phosphate, and 3rd carbon of same pentose sugar making bond with another phosphate. So konchu nalla paathuko inge. Okay, so இப்போ one pentose sugars third carbon and fifth carbon making bond with two different phosphates and making making phosphodiester bond. Okay, and here between the two nitrogen bases, no hydrogen bonds are there. So that's why here we have mentioned. Next one, here you see nucleotide. Simply here nucleotide means three components together we will be considering as a nucleotide, no. So that's why here pentose sugar is there, male P is there, no phosphate. Between the phosphate and pentose sugar phosphodiester bond, actually here we have written phosphoester bond, it is the correct. రెండుమే together na phosphodiester bond we can write. Emma. Next one, between the pentose sugar and nitrogen base, no, what is the bond is there? Inge line is there, no, ఇది glycosidic bond. Emma, ఇది glycosidic bond. And here is equal to is there, no, is equal to ఇది is equal to illama, ఇది enna ఇది hydrogen bond. Here to write a hydrogen bond I don't have place that, but through my voice you can save. Emma, so here this particular point only here we have written. Sorry, ma. So whatever may be that mode, no, my students must understand this. Sorry, ma. Right. Next one, next case, let us enter. Adi enna is side heading. Side heading enna dynamic state of body constituents. So with blue color I have written, no, it is not appearing clearly, but through my voice you can get that this is dynamic state of body constituents. Their subheading is concept of metabolism. That is his concept of metabolism. So there, what is the concept of metabolism? So there, what are the main important points to be discussed? Konchu information only now in this video we are learning. Somewhat information remaining is there. That one I will give you in next video. Now let us start to discuss the concept of metabolism. Now, living organisms from the bacterial members. So from the bacterial members means our scientist, according to the Robert R.H. Whittaker, 5 kingdoms are there, no, ma. So bacteria means Monera, Protista, fungi, plantae, animalia, likewise. So means here there are various types of living organisms are there, no, from the bacterial members to till highly developed organisms, no. So here from the bacterial members to highly developed organisms definitely contain thousands of organic compounds. So suppose if you take one particular living organism, no, we can find out more number or so many kinds of organic compounds. And if you take from the prokaryotes to eukaryotes, definitely we can find out, actually they have mentioned thousands of organic compounds, but more than that only we can find, even in lakhs also we can find out. So thousands of organic compounds. So next one, these compounds or the biomolecules are present in certain concentrations. Means here what is the point, no, here thousands of organic compounds we are taking, no, we are studying, no, there if you take a carbon and if you take a hydrogen, if you take oxygen, so concentrations will be changed in depending on the type of the organism. Suppose if human beings and plants, if you take the, no, plants only either one kind of organic compound will be concentration will be more, and in organisms, no, another kind of organic compound concentration will be more. In plants and animals, no, same kind of organic compounds will not be there. Concentration point of view, so percentages will be vary. So that is my point. Now here, these compounds or the biomolecules are present in certain concentrations. Now, one of the greatest discoveries ever made was the observation that all the biomolecules have turnover. So means here one important phenomenon or discovery scientists discovered. So all biomolecules definitely will have, definitely having turnover. Sir, turnover idi enna sir appadina see meaning enna. So this means that they are constantly being changed into some other biomolecules and also made from the some other biomolecules. அப்படின்னா பாயிண்ட் புரியுதமா, டர்ன் ஓவரான சப்போஸ் ஒன் பயோமாலிக்யூல் இருக்குமா ஆர் சம் அதர் ஏதோ ஒன் பர்டிகுலர் ஆர்கானிக் மாலிக்யூல் இஸ் தேர், அது வில் நாட் சிட் at one place constantly. No, I will not change, so I will be like that only. Likewise that particular organic compound will not sit. So that one all the time being constantly changed or constantly being changed from the one form to or from one biomolecule to other kind of biomolecule. So that is the thing we will be considering. Our subject point of view we are considering as turnover. Amma point purida. One more time you see, turnover means, that means that they are the biomolecules or organic compounds are constantly being changed into some other biomolecules and also made from other some other biomolecules. Means what are the compounds are there, these compounds are changing into another form and some other compounds are being constantly changed into the some other kinds. So that is the thing. So now here what is the main important point here we are observing means this breaking and making is through a chemical reactions or the chemical reactions constantly occurring in a living organisms. So sorry, we see no one chemical organic compound is there means that being changed, constantly being changed, we see no adi because of breaking and because of making. Breaking we see no catabolism and making means anabolism. So that particular chemical reactions constantly occurring in a living organism. So altogether we are considering as metabolism. So metabolism is nothing but the combination of breaking and making. So breaking we are considering as catabolism and making no anabolism. So ipo anabolism and catabolism rendume together we are considering as metabolism. இப்போ டர்ன் ஓவர் மீன்ஸ் புரியுதா, என்ன பயோமாலிகல்ஸ் இருக்குன்னு இன் அவர் பாடி எல்லாமே வில் நாட் சிட் எட் ஒன் பிளேஸ் சிம்பிளி, எல்லாமே வில் பி சேஞ்ச்ட் கான்ஸ்டன்ட்லி. அதுதான். So this is a small information concept of metabolism. Still somewhat information to be discussed in the next video I will discuss. So completely in this video what is information we have discussed, somewhat information of concept of metabolism we seen and diagrammatical explanation of DNA we seen and what are the characteristics that we are observed or that we have seen in B type of DNA also we have seen. So in the next video definitely I will come with next information, continuation information. Until then watch this video clearly and be safe at home. Thank you.